Awesome. Hi, everyone. I'm Sydney. I am a PhD student at UNH with Dr. Nathan Fury. So I will be talking today about a relatively new project with the Marine Biodiversity Observation Network, MBON, and one aspect of that project, which is tracking Atlantic cod movements in coastal New England. So the MBON project is a pretty large project with the primary goal of measuring local marine biodiversity in our coastal waters. And that's using several different metrics of biodiversity, including eDNA, passive and active acoustics, as well as telemetry. And the purpose of this is to try and get at the linkages between marine biodiversity and two of our favorite local predators, Atlantic cod and common terns. And you'll hear much more about the common terns shortly, but I will be talking today about Atlantic cod, my project. So the objectives of the cod project within MBON is looking at movement. And to do this, we're using acoustic telemetry. We're also looking at diet analyses using stomach contents. Acoustic telemetry is done by surgically implanting an acoustic tag, which you see an example of here, which is heard on acoustic receivers, which we have deployed in two different study sites. Our two study sites are in Casco Bay in Maine and in the Isles of Shoals in New Hampshire. Each study site has 12 receivers, as well as two passive acoustic receivers, so hydrophones, as well as an eDNA transect, which you see in red dots, which Dr. Allison Watts is kindly helping us with, as well as active acoustic transects. The crosses that you see depicted up there represent locations where we tagged cod and released them back into the water. So we're just starting to delve into all these data, but I'm just going to point out a couple ways in which we've started to look at movement. What you're looking at here is movement in our main array. Each tag ID corresponds to a tagged fish, and you're looking at detections over time in the different receivers. So what I was kind of looking at here is how kind of site fidelity, how fish kind of stay at a particular receiver, or how much they move between receivers. And you can see there's quite a high degree of site fidelity, and movement that does occurs later in the season, which is interesting. And we can start to look at this in relation to other data that we collected. So this is temperature data that is collected at the receiver level, which is kind of cool because that is kind of a microhabitat level measurement of the environments that these cod are experiencing. And as they're somewhat sensitive to temperature, this could be really informative to our understanding of how cod utilize their habitat. So these are some of the next steps of this project, looking at quantifying the actual movement within each array, as well as integrating the movement data and the diet data with many different metrics of biodiversity, individual level morphology, and environmental data. And fieldwork is expected to continue for 2 more years, meaning we will have a pretty robust data set by the end of this project. So I'll leave it there. Thank you so much.
Hi, I'm Tessa Corsetti Gibner. What kind of lifespan do you have on those receivers?
So it varies depending on how big the tag is actually. And that varies based on how big the fish is. So it's anywhere from a couple years to up to 8 to 10 years. Yeah, and it also depends on the settings that you select as well.
Yeah, it's a really nice talk. I'm curious, if I swim it really hard, it seems like there's potentially interesting patterns before and after the hurricane. What do you think would be going on there?
In terms of the movement?
Where they're appearing and like where they.
Yeah, so definitely have to dive further into the actual data for that, but it could be kind of a, habitat result of kind of that heat wave breaking. We had really high temperatures right before the hurricane, and then presumably a lot of water mixing that occurred after, during the hurricane and after. So potentially some changes in temperature, or, yeah, definitely lots to delve into there.
All right, thank you, Sydney.